Hi guys, so today we'll see how a gene called SDR42E1 helps our bodies absorb and use vitamin D. Along the way, we'll decode mRNA, enzymes, and even a CRISPR experiment. First, let's see how DNA sends text messages over to the cell. So imagine DNA as a school's library master textbook. When the cell wants a recipe, it photocopies a single page. That's mRNA. And it then hands it to the ribosome chefs who cook up a protein. And speaking of proteins, let's find out what they actually do. Proteins are the cell's toolkit. Some build, some move, some signal. Proteins are made up of a chain of amino acids. Enzymes are a special class of proteins that speed up chemical reactions inside our bodies, like adding rocket boosters to ordinary tools. But how do enzymes hit the gas? Let's take a closer look. Picture an enzyme as a lock and its target molecule is called the substrate, a key. Only the right key fits. Once locked in, the enzyme bends or splits the substrate, turning it into a product at a much faster pace than normal. Now that we know what an enzyme does, let's clear up another term: vitamins versus hormones. Most vitamins are nutrients that you eat, while hormones are chemical messengers your body makes to broadcast instructions. Vitamin D is both. We get it from sunlight or food, and then our body converts it into an active hormone that talks to cells. But why does that matter? Because vitamin D keeps us healthy in big ways. Active vitamin D helps your intestines pull in calcium for strong bones and tunes your immune defenses so they don't overreact. Low vitamin D can mean fragile bones and sluggish immunity. So where does vitamin D come from, and how is it switched on? Sunlight turns a skin molecule into vitamin D3, but that raw form can't talk to cells yet. First, the liver adds on a hydroxyl group, making 25-hydroxy-D. Then the kidney adds a second hydroxyl group, producing 125-dihydroxy-D. This is a fully switched-on hormone that binds the vitamin D receptor and changes gene activity. Now let's meet the gene that helps us absorb vitamin D in the intestine. SDR42E1 makes an enzyme that lines your intestinal wall. Think of it as a friendly bouncer opening the door so vitamin D can enter the bloodstream efficiently. But what if we deliberately break that gene in the lab? Using CRISPR, scientists cut the SDR42E1 gene and introduced Q30, a stop codon mutation. What that really means is that instead of a normal 300 amino acid enzyme, the mutation prematurely stops the assembly of amino acids, and a 30 amino acids long, useless protein is produced. This stunted fragment can't function as an enzyme at all. What does losing that activity do to a cell? Mutant cells took up far less vitamin D and showed stress signals. To understand the full picture, the team opened their multi-omics toolbox. Omics just means any technology that studies everything in a category. Like all gene study is called genomics. Study of RNA is called transcriptomics, or protein is called proteinomics. I wonder how the scientists do the study. First, RNA-seq revealed how 20,000 genes dialed up or down. Dialed up simply means the gene's activity. Its mRNA output increased, and dialed down means that activity decreased. Then, proteinomics, mass spectrometry that tallies every protein molecule, showed real-world changes in 5,000 plus proteins. Altogether, 4,663 genes and 140 proteins shifted. But raw lists are messy, so scientists grouped them into pathways. What exactly is a pathway, and why do cells need them? A biological pathway is a cell's assembly line. Each molecule passes its product to the next. Cells use these routes to respond to signals such as nutrients, hormones, light, temperature changes, or stress. Now, which of those traffic lines jammed when SDR42E1 failed? Sterile metabolism tracks sped up. Transfer proteins LRP1B and ABCC2 fired into overdrive, and the Wnt cell cycle pathway lost stream. That's a lot of things happening, but what you really need to know is that together these shifts mess with the nutrient handling and cell growth. I wonder how badly this cell suffered. With SDR42E1 disabled, only 47% of cells stayed alive after 72 hours. Meaning half the intestinal lining would wither if this happened in a person. Such low survival can impair nutrient uptake and trigger inflammatory damage. Re-adding SDR42E1 lifted survival back to near normal, providing the gene's protective role. Some tumors overactivate Wnt and sterile pathways to grow. Restoring normal SDR SDR42E1 activity in a healthy tissue may prevent that hijack. In contrast, if a tumor already depends on SDR42E1, drugs that block its enzyme function could starve the cancer, based on the tumor whether we decided to boost or break the gene. Vitamin D deficiency affects over 1 billion people. If some cases trace back to SDR42E1 variants, genetic tests could flag who needs higher vitamin D or future enzyme-targeted therapies. Let's wrap up with a quick recap. Here's what we covered today. mRNA turns DNA plants into proteins. Number 2 is that vitamin D acts as both nutrient and hormone. Number 3 is that SDR42E1 helps intestines absorb vitamin D. Number 4 is that breaking SDR42E1 hurts vitamin D uptake and cell survival. Number 5 is that this insight could guide treatments for deficiency or cancer. Thanks a lot for joining us on today's video. We hope you had a lot of fun and learned something new. If you like this video, don't forget to like and subscribe for more Biology Bites. If you care about helping other kids learn, check out Pratham USA. They bring reading and learning to children across India and help build a better world. Visit prathamusa.org to see how you can help. See y'all next time. Bye!